So nice to meet you. And my name is Ana Maria Heilman Morales, and I am the team leader of the Agricultural Experiment Station Big Data Pipeline Unit. I am responsible for overseeing the development and the implementation of data management and the analytic strategies that support this Agricultural Experiment Station. And I am really happy to be part of the National Association of Plant Breeders. And I look forward to sharing the information that we have and our expertise with you. And thank you for having us today in APB. I would like to introduce to our second speaker. Our second speaker is Didier Murillo Flores. Didier, you can say hi. And Didier is our statistician developer. He's affiliated to the NDSU Big Data Pipeline Unit. And he has been working with our team for the past 3 years and a half. Didier is leading the development of our Shiny tools, and he is the main developer of FielDHub, which is a click and point type of application that has been built in our Shiny, and it helps to streamline the generation of randomizations as well as maps for the field. Our third speaker is Johan Aparicio Arce. Johan, please say hi to everyone. He's a very talented statistician as well as Didier, and he is affiliated to the International Center for Tropical Agriculture, which is located in Ciudad in Cali, Colombia. And he has been working for the past 5 years as a statistician for the dry beans breeding program. But currently, he have expanded his role and support different teams, especially on the data consolidation, the data analytics, as well as the software development. Johan is the main developer of MrBean, which is also an easy-to-use or Shiny application, and it helps in the simplification of the analysis that for those large-scale plant breeding or agronomic or the biological experiments that it can be used for that as well. And it uses the power and versatility of the linear mixed models. For the past 3 years, we have been collaborating with Salvador Guesan. Dr. Salvador Guesan has been very important for us to be able to integrate ASRMLR codes. And also he has been very important in supporting our understanding, like of different, concepts that include, like a multi-environmental trial analysis, single location analysis, and some of the information that we are sharing with you today. So thank you for joining us. So in this second part, we are going to be sharing with you some of the experimental designs that we didn't mention in our previous session. Some of them for the FielDHub are the unreplicated designs, the PREPs, the augmented experimental designs. And then we are going to show you a new capacity that we have in FielDHub, that is the sparse testing allocation for multi-environment trials. We are also including a new feature that is the average allocation in partially replicated designs for the multi-environmental trial analysis. And we are going to, do a little brief, concepts about blobs and blues. Then we are going to go with MrBean. So then we are going to do design of experiments as we did last time, and then we are going directly to the analysis of the data. In this time with MrBean, we are going to talk about the multi-environmental trial analysis on the 2-stage, the G/E interactions, and mentioning the factor analytics. We also are going to show you a new tool that has been integrated within MrBean, that is the gblob. And then we will go with the conclusions and the questions. So as I was mentioning before, so this is the different people that has been able to make this possible, or it's a collaboration. And then, as I say, Didier is the main developer, but this is all the people that have made contributions to it. So let's dive in and let's talk about unreplicated designs. So unreplicated designs are also considered or called unrep designs. And then we are talking about a specific type of design that is the one that contains checks on diagonals. So these type of designs are characterized by the assignment of the treatments or the genotypes to experimental units that are unreplicated within each environment. So then when do we use this type of designs? Well, when we have, let's say, a large numbers or large population sizes, and then the treatments, we don't have maybe a lot of seed for them. That's the case of the preliminary yield trials. Also, when we have, let's say, large experiment blocks. So at the commercial level, let's say that we have blocks that are bigger than the 250 treatments, and then they can be arranged systematically in a manner that you can test a lot of experimental units across different locations. Then the other one is that those checks or those control plots are going to be assigned. That can be two ways. Well, this one that we are going to show you today is going to be in a systematic manner, but we are also going to show you in a random manner. But the reason why we use those control or the check plots is because we want to, estimate using those replicated checks, the experimental error for those unreplicated ones. So then this is an example of how a field looks, or a field layout looks in FielDHub once you utilize this type of experimental design. So some considerations to have in mind when you are using this type of designs is that you need to carefully select the checks. And then we are going to talk about, like the performance checks as well as the quality checks. So then the actual arrangement of those check plots is not as important as the number of checks or the check plots that are in the field. And why is this? Because, it helps us to estimate better the effects of those genotypes that are unreplicated. So what we suggest is if you are going to utilize this type of experimental design, do not use less than 8% of checks in the field. The more the better, but then also this experimental design is better when you have multiple locations. So as I say, it is appropriate for large experiments that have more than 250 genotypes that are tested in multiple locations and that are arranged in big decision blocks. Medium to small experiments are not the best. This type of design wouldn't be the best. So let's talk about how to select a performance checks. So these performance checks can either be a variety, a known variety, or hybrids that are used to assess the reliability of those yield trials. So these standard varieties usually are chosen based on their characteristics such as the yield potential, the disease resistance, the maturity, and they are included as a benchmark for comparing the performance of the experimental units. Then this is very important to have in mind to control the heterogeneity of experimental units or those field plots. Replicated checks must be distributed over the field experiment area, where the actual arrangement of the check plots, as I said before, has less importance than the increasing number of checks. So control plots are effective if the yield or another trait that is being tested of the checks shows the same general pattern response across the trial as the test genotypes. So now in here, we give you some examples of the arrangements or the field layouts. So then we are going to show you the diagonal arrangement. That's the one that is kind of some of our breeders prefer these arrangements. And the reason why is because they go to the field and they can see the checks in the field. So that is kind of something easy for them to identify. There is the other option that is the completely random. And this is where I'm going to, stop. And please, Didier, go ahead.
Yeah. Hi, everyone. Anna, do you wanna explain the same example? Okay.
Sure. Do you want me to explain the single and replicated experiment?
Yeah, if you want,
okay.
I can do the demo.
Okay, perfect.
Yep.
Sounds good. Let me see if you can see my screen.
Yep.
Perfect. So then in this example, we have, let's say, a single and replicated experiment in which we have, some entries of barley. Let's think about barley, right? Then we are going to set up this PYT experiment. Let's pretend that we have 300 genotypes. And these 300 genotypes, we are going to put them in an unreplicated experiment. Then, as you can see in here, 300 is shown kind of let's pretend that in the field we only have the ability to lay out 15 by 22. That means 15 rows by 22 columns. sometimes like one of the cool things about our tool is that it allows you to specify the dimensions of that field, and that helps you to map that particular experiment. So then in this case, we want to use 5 checks, and I don't know if you can see my little mouse, but then you can see that the checks are put in with numbers 1 through 5, and then they are scattered through the field, but they are systematically arranged. And also you can see that like we have about 30 plots of those checks that represent 9.1% of the total number of the experimental plots. So then that is an example of a single unreplicated experiment. So there is another example that is called the stack or the multi-unwrap experiments, right? Now we have the same breeding program. It has 300 genotypes, but then let's say that those 300 genotypes, you know, like we want to split the genotypes that are being evaluated for different, you know, reasons. And we have an experiment like contains the 100, you know, entries, another has 120, and the other one has 80. So then we are creating a block in the field that contains those 300 genotypes, but then we just want to divide them in 3. So then each experiment represents, you know, like let's say stages of maturity. But in this case, I would say like we can use what we can identify like a different, you know, like an objective. So then what we can do is specify in the program, okay, I want to have a multi-unwrap experiment, define the rows and columns. In this case, that's the 15 by 22. And then we have the 5 checks allocated throughout the field. And same thing, we have 30 plots that represent 9.1% of the total experiment plots. Now, okay, so then this is the part of the augmented designs, Didier. This is where you want to start, or do you want me to stop in here?
Yeah, that's fine. If you want to stop, I can keep going
Perfect.
from here. Yep.
Yes, perfect.
Thank you so much.
Yep.
Okay. Hi, everyone. I'm Didier. Thank you, Anna, for the kind introduction of unreplicated designs. The explanation was great. Augmented design are another type of unreplicated designs where we are adding a new structure that is like a blocking in this case. But blocking the blocks, it can be as well the rows in the field or the column in the field, or another type of blocks, like for example, that we know as blocks in a randomized an RCBD design, right? For this experimental design, we have to take care about like the control plot, that they are the checks. They are replicated using a standard type of experimental design, for example, an RCBD. And also we have some treatments that we can consider like a new, like it can be genotypes that you want to test and evaluate in the field. We say, for example, we have 4 checks in a RCBD design. Those checks are replicated across 6 blocks that we call that augmented blocks. And then the new treatments augment the RCBD design that you can see in here. Like we have the checks, for example, in here, I don't know if you can see my mouse, but the check 2, the check 1, the check 4, and the check 3, they appear in the first block, in the second, and so on, up to the last block. And also we have the new genotypes of the treatments across the entire block. What is the difference with a regular RCBD? In an RCBD, all treatments appear in all blocks. In here, no. The treatments or the genotypes that we have in the blocks, they are different. Like that is the main difference with the RCBD. The only that is in all blocks are the checks. After the introduction of unreplicated designs and the augmented design, just I want to mention some statistical consideration that we have to care about that, about those designs when we want to analyze the data. When analyzing replicated designs, it's important to keep in mind that those designs have some limitations. For example, for the lack of replication is a limitation of those designs. To account for this limitation, we recommend to use appropriate statistical models, for example, or a statistical method. For example, we can use linear mixed models with autoregressive spatial structure, as well as spatial models that include a smoothing method like splines. Why we recommend that? Because if you, we know that it's a lot of papers that talk about this, but the truth is in the field, we have a lot of variability and a spatial correlation. That spatial correlation refers to the tendency of nearby experimental units to have similar responses, which can affect the estimation of the treatment effects. This can be, for example, this image, like a visualization of those spatial correlation. After we know the basics about unreplicated design augmented, just I want to introduce you FielDHub and how to make those randomizations using this tool. For be able to use in FielDHub, we only need RStudio, and we need to install the package because it's as well as another package. The way to get the application, you can say install that package, FielDHub. I won't do that because I already have the package, and also some of the features that I will show, they are not yet in ArcRAN. They're going to be a release for the next week. Yep. But for use, the only thing that you say library, after install the package, you can say FielDHub, and can say, for example, FielDHub, : run up. And after that, in your preferred browser, it's going to open a web application that is FielDHub. We have different modules. We have the modules for unreplicated designs. We have a module for partial replicated designs. Also, we can do lattices. That this was like the 1 week ago, we explained, we show how to make that. And also we have another type of experimental designs, like the classical ones, like the completely randomized design, the randomized complete block design, Latin square, factorial, split plots, incomplete block designs, and resolvable row column designs. Yeah, but for today, just I want to show you how to randomize, how to create these designs, this type of unreplicated designs. The first option that I want to show you is, remember the example in the slides was 300 genotypes and of barley, and we allocated 5 checks. Yep. Okay. In this here, we can set, okay, the number of entries that we want. After that, you can select the number of checks that we want to put in the field. But before that, also you can say, if you already have the list of the treatments with the number of entry and the name of that genotype, you can upload that in a CSV file. The requirements that file should have one column with the entry and another column with the name of those treatments, including the checks in the first rows of the file, right? But for today, just I want to show how to make it, generate the randomization using data inside of the app. After that, you can select how many locations you want to randomize this experiment. We say just 1 location for now. right now you can say in the field, you can set in here like the way to set up the planting, the plot number. You can say, for example, serpentine and Cartesian, let's say serpentine. Also, you can set up the seed number. The seed number is, I would say that is pretty important because we eventually, maybe we can recover. We want the same randomization. And then you can use that seed number for that. For example, let's say that 23, in a couple of months, I want to make the same randomization with these same parameters. I can use this number and the results, the randomization is going to be the same. Yep. You can set out the name of the experiment, for example, barley 2023. And you can say that it's the plot number where you want to start, for example, 1001. And you can set the name of the location that you want to allocate those. After that, you can run that. And the application is going some calculations. And after that, you can select the field, the dimensions in the field that you want to deploy your experiment. Let's say that I want to 15 rows, and this is the option 15 by 23. I like that. And after that, you can randomize. Now we have a table with some information. Remember that we need to allocate some checks in this type of design. And this column is telling you, okay, we have some percentage of the checks. You select one of those, and based on your selection, you are going to allocate, for example, if you put 6.667, and you are going to allocate 23 checks. But for completing or for fitting the dimension in the field, the application is going to allocate as well some filler plots. I don't like that too much. I like an option that we don't have to put too many fillers, just one, and that is, it can be 12.75% of checks. That is equivalent to 44 check plots, and just want to put one filler, right? And then randomize and go to here, and this is the field. And now we have the information of the columns in the field, just one single filler in here. As you can see, we have the checks in a systematic way, that is a diagonal, and all the checks are replicated with this frequency. You can play a little bit with the percentage of check-ins here. You say, oh, I don't want too much checks, maybe a little bit less. We'd say, for example, 11%, and now the field is looking in this way. Also, we have the plot number. Remember that the plot number is going to start on 1001 and is going to serpentine. What does mean? That is, the row ends in 1023 and starting here automatically. That is serpentine. And finally, like a very important file is the field book, where we have the name of the experiment, the name of the location, the year, the plot number. We have the information about the row in the column in the field, and we have a column that whether the treatment is a check or not. This is important for eventually you want to analyze the data, and you want to like split those checks on those treatments in your model. Also, we have the name of the entry, and we have the name of the treatments. If I go here, just I want to put the maximum percentage checks, and that's it. Sometimes some users of FielDHub, they are professors, teachers, they teach statistics using that. You can simulate some data. Let's say, because this is like an spatial randomization, you can put some correlation into rows and put some correlation into columns, and set, for example, okay, I want yield. You can set out another traits, but for now we say yield from 45 to 101. And let's go. And now created a new column for you with the response variable. If you want to see this, you can see in a heat map. This is a heat map like showing the variability in the field and other things. Yep.
Didier, we have a question in the chat.
Yes.
Somebody's asking, did it work for all crops? And what is the percentage of error while using this hub? So maybe I can start with the first part of the question. So did it work for all crops? I think that this is best suited for field row crops, in which you can, like put these type of designs, especially in the field. The other part of the question is, what is the percentage of the error by using this type of experimental design? Go ahead, Didier.
That depends. That depends because just like Anna said, we need this unreplicated design have some limitation, yeah. And the only way that you are estimating the experimental error is through the checks. Yep. If your checks are not good, if your experiment is a small 100 genotypes, or your uncertainty is going to be huge. Yep. But if you put a good amount of treatments and a good amount of checks and a good checks, your uncertainty and the prediction is going to be lower. Yeah, I cannot say like exactly what is going to be the error, but I can the thing that I would say is you need to consider some things to make this better. I don't know if that answered your question. Okay, this is the unreplicated. Also, we can do this, like allocate multiple experiments in the same. Let's say we have the same 300 genotypes, and you want to split those in 3 blocks can be, or experiments, sub-experiments, and I want to put 5 control plots. The other thing that you already know what is going on with that. The only difference is that I want to show you the difference. Let's say I want to, in this way, and if you go here, this is the, yeah. And after that, this is like the layout for your experiment. You have the experiment 1, the experiment 2, and the experiment 3. Also, we have the plot number, and we have the field book. If you don't want this like a setup for the layout of your experiment, maybe you can put it side by side better. You can select here like a by column and run again. And for example, this again, randomize, go here and go here. And now you have like you set up 3 experiments in the same field. As well as if you want, you can simulate some data. In all of this design, you can save your experiment. And let me show you. If we go here, what is this? Okay. Yeah, we have this is like a field book, the experiment location, year, plot. You can download it to CSV, to Excel file, and save it, for example, in your database or anything that you want to do with that file. Also, we have
sorry for interrupting.
Many questions.
There is one question.
Yes.
Yeah. Yeah. Go ahead, Anna, if you wanna.
No, go ahead, Johan.
Okay, yes. If the experimental field, for example, 300 genotypes was divided into 3 blocks of unequal size, does this software work?
Yes. No, it can be the equal side. I can put 100 and 100 and run again. Let me show you this.
Yeah, and in his example, he was saying 100 and 149 and 51.
Yeah, can you see? And now we have equally sub experiments. They want to set up 100.
149 and 51.
100 and now 149.
49,
And which one more?
51.
51. 51 is pretty small, but let's try it. Yep, the smaller one at the end. And the application is going to handle everything for you. Yeah, you just set up these numbers, the sum of these. If you sum all of these, it should be equal to 300. That is like the main. If you put 301 in here, the application is going to scream to you that error, number of treatments in block that doesn't match with the data input. Yep. Any other question?
So far, so good.
Okay. Let's go for the, just like Anna said, that you can set up the unreplicated designs in other ways. For example, let me show you just for like in a randomly allocated into the field. What does mean that? In this case, we are not putting like the restriction that the control plots should be in a diagonal system or in a systematic system. Yep. All of those designs allocate the checks in a way that in a such way that all the columns in the field and all the rows in the field has control plot. Because remember, eventually when you go to a field, deploy these experiments, and those rows and columns, maybe they are going to end up with correlations. Yeah. And we need to use that information for estimate those correlation and get your predictions. For that reason, it's very important that the control plots, they should be spread in a good way in the field. Also, we have the field book that is like the other part is the augmented RCBD. Exactly, we have, let's say we have 50 treatments, we have 4 checks, and we can set up some augmented blocks for those this experiment, and you can run. And after that, you can select the dimensions in the field. For example, I want to say that I want this. You got some information about the randomization, and this is like the layout. Yeah, this is the plot number, and this is your field book. And that's it. I don't know if we have more questions because I would like to move on into the sparse allocation theory a little bit.
So far, there is no more question in the chat, so you can go ahead. There is one question.
Okay. Can you read it, Johan?
Yeah. Can this also be applicable in regular experiments like RCBD with less than 10 treatments?
If your question is that you can use FielDHub to make that RCBD, yes, absolutely. But you can use it RCBD in here to generate 10 treatments.
Yeah.
Yep. You can generate an RCBD in here with 10 treatments. But if your question is use unreplicated with this type of, my answer is no. No. No.
A person wants that you could demonstrate for multiple locations with 300 genotypes. So would it be possible to go into the unreplicated design? Are you meaning the unreplicated designs?
Okay, yes. How many locations? Let's say, for example, let's go to a single diagonal arrangement that is in the diagonals. Let's say 300, 5 checks, 5 locations, or we can put maybe 10. And let's go. Now the application is doing the calculations, and now we need to select the dimensions in the field to set up the experiment. Let's say that I want this. Yep. that is 14 rows and 24 columns. And then after that, randomize. And if you can see, we can put 10.71% of checked. That is equivalent to 36 checked, 0 fillers. And then go here. And this is the randomization for the location number 1. If you want to see the other location, you can go here. And for example, I want to see the location, the randomization, the location number 5. And now that should be different because the randomization in location, they should be different. Yep. And after that, the field book. In the field book, we have 1 location to 10 location. If you want to see, for example, the field book for the location 7, just filter here, and now we got the information for the location 7. Yep. If you want to simulate data, it's already in. For each location, you can see as well. If you want to see like a heat map for the location 2, that is going to be different. Can you see some correlation in this part, like some variability in the field?
And this part is more like kind of like for making sure that people is understanding the idea of correlation, and also kind of like to play with the tool as a teaching.
Yep, exactly.
Resource.
I see that we have a lot of questions. That is good.
We have another question in which he's asking, sir, if the blocks have unequal dimensions, will it work?
What is the question? If the blocks have
unequal dimensions, will it work? Yeah, we show the example with which you have.
But in which design? In the
thing is unequal dimensions. You know, like you mean like you have an experiment with, let's say, a 12 by 15 or whatever, and then another one that has what, 10 by 11. Is that what you're talking about? We can continue and then we can go back
at the end.
To that question is at the end.
Okay. This is a very interesting topic that we can use in MrBean, that is called in agriculture, that is a sparse allocation. What is a sparse allocation? Basically, this method allows for the allocation of genotype treatments in a such way that not all of those treatments are grown in every environment. In the experiments that I show you, for example, it was a pretty good question that set up that randomization across with the same 300 genotypes across multiple locations. But when we set up the experiment in that way, we are getting replication for those 300, 1 replication into each location. If we have, for example, 10 locations, we are going to end up with 3,000 plots, right? But maybe sometimes we want to allocate into those locations, but we don't have a lot of seed. And maybe we can decide, okay, I want to put this genotype in the location 1, 2, 3 until the 9th, but I cannot put it in the last one because I don't have seed. Yep, that is like basically a sparse allocation. This approach is often used when seed resources are limited and can be potentialized by integrated with this method with genomic relationship matrices. Yep. Just I want to use a simple example, a toy example for showing you what is a sparse allocation. Say we have 5 genotypes to allocate into 6 environments. And if you do this, like solving this problem, with this problem will end up with not all genotypes present in all locations. Let me show you. Let's say we have an environment 1, 2, 3, 4, 5, and 6. And we want to allocate those 5 treatments into that 5 6 environments. Let's say, for example, the genotype 1 is going to be present in the environment 1, but is absent in the environment 2, but is present in the other ones. What's happened with the genotype 2? Okay, it is present in the first specify environments or location, but it's not in the last one. What is going with the next one? Okay, it's not in here, and this is a sparse allocation, and I want to go to some advantages of that. Like if we use in a sparse allocation, we have the possibility of increase efficiency. By using a sparse allocation, breeders can evaluate a large number of genotypes or treatments in a single experiment across multiple environments, which can accelerate the breeding program and reduce the time and resources needed. Another advantage is selection intensity. What does mean that? The large number of genotypes or treatments evaluated in a sparse allocation experiment can increase the genetic diversity in a breeding program and increase the chances of identifying superior genotypes or treatments. Another one that is also pretty important is cost effective. Sparse allocation experiments are generally less expensive compared to a full replicated experiment, since fewer experimental units are needed. Yeah, maybe we don't need to allocate the 3,000 plots. Maybe we only need 2,500, right? And we can save a lot of work and money. But as we have some advantages, we have some disadvantages. Less accurate predictions, yeah, because the limited number of experimental units means that the estimates of the treatment's effect are less precise compared if, for example, those predictions can be less precise if you compare to a fully replicated design or an unreplicated that you replicate in the same way to many locations. Yep. But however, an increase in selection intensity may compensate for the loss of accuracy. That, as you know, that is a trade-off problem. It's a decision, but you gain on one side, you lose a little bit in the other side. Another disadvantage is difficulty in identify environmental effects. Since not all genotypes or treatments appear in all environments, it can be difficult to identify environmental effects on the performance on the genotype. Yep. And now I want to tell you how we can generate that. It can be generated using different design strategies. We will talk about the random allocation and using incomplete block design idea, like the principles behind incomplete blocks. Let's say that we want to use random allocation. It's pretty easy. We can do it even maybe in Excel if you want, but the random allocation, it is simple and convenient way to generate an sparse allocation design, but it may not be the most efficient or informative approach. The allocation may not be balanced or representative of the design space, which can lead to less precise estimate of the treatment's effects. The other approach there are many, but for today's use, I want to talk about those two. The other approach is incomplete block designs. In this case, we are using the principles or the theory behind of incomplete blocks to generate the distribution between locations. This design strategy allows for efficient evaluation of a large number of genotypes or treatments while maintaining some degree of balance. Just I want to read this quote from this paper. IBD for the incomplete block designs for the allocation of lines to location can help improve predictive performance compared with random allocation. This has the potential to be applied to a large-scale plant breeding programs. Maybe I can do the demo of this before to go to the partial replicated design. If you go to unreplicated design, this is a new feature. If you download FielDHub right now from CRAN, this feature is not available. Next week it will. And now if you go here, the first step is set up the number of treatments that you want to allocate into those locations. After that, you can set up like the way that we want to allocate the checks in the field, the distribute in the field. It can be diagonal or random. For this case, let's say diagonal. After that, you will need to say the checks. Remember the checks in a replicated experiment, we need checks. And also the locations. Let's say we want 5 locations. And this part is very important. This the number of copies per plant. If we have, for example, 5 locations and we want to allocate 4 copies per plant, what does mean? That some location is going to be, some genotypes is not going to be in some locations. Yep. After that, let me run this. It's doing the optimization and it takes a few seconds. Okay, after that, we have again the dimensions that the application suggests to you to make the randomization. And also we have this allocation table. When the rows, we have the genotypes and the columns, we have the locations. As you can see, for example, the genotype 1 is not present in the location 1, but is present in the other location. If you see the genotype 2 is not present in the last location. You can download this. We have in here, and you can play with it. After that, if you want to randomize in this way, you can randomize and go to there. And now we have the randomization for the sparse location, using a sparse location to 5 locations, using 4 copies of each genotype when we want 380 genotypes using diagonal. This is for the first location. We have, like at the last genotype, I put it automatically, the application to set the check plots. And you want to see the second location, for example, and so on. And also we have the field book in here. I don't know if we have questions so far.
No,
Nope.
no.
No? Okay. Liz, yeah, this is like a sparse for unreplicated designs that it was a little introduction, many things behind. But the important thing that I want to show you is how to make those sparse allocation using FielDHub. That is is quite easy. Yep. If you want to save the field book, you can download it. You can simulate data. But for my last part, just I want to talk about a little bit about, okay, some comparisons. Yeah, I was talking about 2 ways to get the sparse allocation. That was incomplete blocks and random. What is going on with the incomplete block? Let's say that we have those locations, and those locations, they are zeros or ones, right? They're a binary problem. If you transpose that matrix of zeros or 1, you can calculate the Euclidean distance between those locations and see what is my generic distance between the location. What does mean that? What does mean that if we have a generic distance that is small, that is good. Yeah. But if that generic distance between location is higher or is quite high, what does mean? That the location or the genotypes are not like a well shared between locations. For example, using incomplete block designs, we can say that it's pretty balanced, and that is good. All the generic distance between location is 0.33 with a little difference with the last digit, but they are pretty similar. This is pretty good. But what's happened if you see random allocation? That is pretty easy to do it, but it's not the best way to make it, to generate the sparse allocation. For example, we end up with generic distance that is, for example, between the location 1 and 2 with 0.32, but for example, in this case with a lower than, another case with a higher. They are different, and that is not good, because when you are going to try to do the statistical analysis, that this unbalance problem is going to generate some issues in the comparisons and in the predictions, and it's not good. Yep. The best way, incomplete blocks, that is the approach FielDHub use. Partial replicated designs, we have, in this partial replicated design, we have a portion of treatment that are replicated 2 or 3 times, as well as the rest of the plots are filled with unreplicated treatments. Let's say an example, we have 22 treatments, and 75 is going to appear twice, and 150 just 1 time. For example, but what's happened if you allocate, or if you replicate the same experiment across many locations, you can call it las augmented p-rep. Yep. The other way to make this p-rep, you can say, okay, I have 5 locations, but I want all the treatments has the opportunity that make at least 2 replications. For example, in the location 1, you have a set that is replicated and a set that is unreplicated, but you don't want that the same set in all locations. Yeah, you want different. And the thing that you can say is, okay, I have an amount of copies that I want to put, or maybe you want an exactly average per genotype across multiple locations. Yeah. And let's say that we have 6 environments, we have 5 genotypes, and we want to allocate 9 copies of each one. Okay, this is another optimization problem. How we allocate this problem? Yeah, you can say, yeah, okay, I know that is 9, but how do you distribute that? You can do it randomly, but also you can use IBD or the concepts behind incomplete block designs. In this case, each genotype has 9 copies across the 6 environments, and you got an average of 1.5 by treatment. Let me do the the demo for this. If you want to make it, go to partial replicated designs and the augmented. If you do it across multiple locations, the first part is you want to define the treatments that you want to replicate. For example, 75, I want to replicate it. And the treatments that you don't want to replicate, for example, 150. After that, you say that this 75 you want to replicate twice, and this just 1. And run this. They show you some options for the dimension of the field, and go. Just I want to show you this. This is the field randomization. Also, we have the plot number, and we have the field book. You can make it across multiple locations, let's say 4, with the same setup. Let's run again and run. It's taking a little bit. Okay, now if you go here, you want to see the location 2. They are a little different. The randomization is different. And yeah, this is like the augmented p-rep if you do it across multiple locations. But what's happened with the optimization problem that we want to allocate? Okay, we can say we have 360 genotypes, and I have the problem that I want to allocate those using a p-rep into 6 locations. You can say that you want to put some checks that some breeders do in this kind of p-rep as well. Let's say that I want to put 3 checks, and I want to replicate each 8 times, and set the number of locations. And now I want to say that I want to 8 copies of each genotype across those locations. Let's run this. It's going to take a few seconds while does the optimization.
they did a lot. And while this happened, I just wanted to mention I have been putting some, resources in the chat so people can go directly to the papers and to the GitHub.
Okay, first of all, the application show the dimensions they available to make the randomization into the field, to deploy the experiment. Yep. And after that, the same table that we use in sparse allocation, but in this case, we don't have zeros anymore because we have more seed, and you can allow to put maybe 2 copies and 1 copy depending on the location. For example, for the genotype 1, there are going to be 2 copies of this genotype or treatment in the location 1, but just 1 copy in the location 2 and 3 and 4, as well as two copies of this genotype in the location 5. We got 8 copies for each genotype and an average of allocation of 1.3. Yep. Let me select this way and randomize. If you go here, it's doing the randomization. And now this is the partial replicated design using the same ideas of sparse allocation as well, using incomplete blocks to make the optimization. And that's it. This is the field book. And if you want to see the location seats, this is different. The treatments that are replicated in the location seats are different, are the treatments that are replicated in the location 1. And so if I think that is good enough, just I want to show, if you want help, we have all the tutorials for the functions, all the thing that I did, you can do it in the command line using R because FielDHub is an R package. But basically we have all the information in here. And also just I want to mention of who is behind of FielDHub. I'm Didier. Dr. Salvador Gesana has collaborated with us as well. Dr. Anna Hellman and Matthew Seifel, Johanna Paricio, and Dr. Richard Horsley. And this has been a collaboration between an international group and to trying to make something for helping people. And I want to say that thank you for everything that is contributed to this application. And I want to mention that FielDHub is like a primary tool for make the randomization, and plant science department, the breeders that they use it in here.
Okay, perfect. I don't know if there are some questions. It seems there is no question, so I'm gonna start from here. I'm gonna share my screen. Just let me know when you can see my screen. Perfect.
Very good.
Thank you, everyone, once again for joining us. So now I want to explain a little bit more about the multi-environmental trial analysis. And at the end, I will give you basically a demo about this methodology. So as we mentioned before, this is more or less, what MrBean is focused on. MrBean is an easy-to-use Shiny web app that simplifies the analysis of large-scale plant breeding experimentals by using the power and versatility of linear mixed models. So it not only helps us to analyze single trials, but also help us to analyze multiple locations. I'm gonna start with this introduction. This is something very common, where basically a set of genotypes are tested in different environments, where basically we know that the cultivar might be exposed in the future. It can be heat stress, drought stress, irrigated conditions, different regions, etc., etc. So basically, that's the meaning of MET trials, when we have many locations, many trials, etc., etc. And the idea with this is basically how are we gonna analyze this amount of information, General uses, we have many locations, we have many sites, we have different conditions. And the idea is to incorporate all of those sites, all of those locations to estimate overall breeding values. And it's also important to, to understand this genotype by environment interaction. So this approach basically allows us to estimate the genotype by environment interaction and also the variant structure, how much is genotype, how much is genotype by environment interaction, how much is error, etc., etc. So it's critical to understand the genotype structure of the population and to define breeding strategies. I brought this graph. I mean, this is the paper where I took it, but it helps us to a little bit to understand more about the genotype by environment interaction. Here we can see on the left side, let's say irrigated condition. On the right side, draw condition or heat condition. And it is called, for example, in this paper, no plasticity or no G by E, where we have basically the same genotypic variance in both locations and both stress, and also we have the same ranking. So there is no plasticity or no G by E. And you can see that the mean in this treatment is the same that the mean in this other treatment. And now if we move to the next one, the plasticity but no G by E, we can see that there are difference in the mean. So this one has a lower mean than this one. Also, the genotypic variance is the same, so it's not too complicated. Then we have this plasticity or divergency, G by E, where we have different genotypic variances. So for this one, the irrigated condition, we have lower genotypic variance, and the other one, a higher genotypic variance. But, the ranking is the same. That means that basically the genotypic correlation is positive, and the ranking is basically, has no change. This one is basically the convergence is in the other way around. But as I said, it's not a problem, The more complicated scenario, I would say, is the plasticity or crossover interaction. We could have different genotypic variability in different stress, but also, as you can see, the red one is the genotype number 1. In the first stress, the first treatment, it has, the worst yield, for example, and in the other environmental condition is the best one. So the correlation is totally negative, so the ranking change totally. So that's why they call it crossover G by E interaction. And this is like a more complex scenario when they say there is no linear G by E. Here I have also an example just to illustrate this. We have 2 locations, let's say Colombia and the other country here, Colombia and Peru. And what if we don't check the genotypic correlation? So if we just analyze, the 2 locations and we just take a mean across the location. So if we don't check first, like the genotypic correlation, we are gonna basically damage that ranking. And this is basically what I'm gonna do with this example. Here, I just detect that there is a negative correlation, but what if I just estimate an overall breeding value across the location? So basically, I cannot differentiate or distinguish between the best one and the worst one. Almost all of them are the same, and they have the same yield in average, That's why, just to make emphasis on this, that it's important to check what is the genotype by environment interaction, or what is that correlation between trials. This is just 2 locations, but imagine if we have 10 locations, 20 locations, 100 locations, etc. It's more complex. Some difficulties: every site has its own personality. When we say personality, it's because is a totally different, environmental condition, even though we try always to have the same environmental conditions sometimes in some treatments. But the point here is that the error is different. Probably there are some management effects that can affect also our response variables. And, sometimes we have some experiments with a different experimental design. For example, in this location, I have an RCBD. In this other location, I have an alpha Alaris, row and column design. So that's why we call it like personality, because all of them are kind of like they have their own characteristics, and we have to take into consideration. So we need to think that needs to be combined into a single analysis. So we need to consider all of these things. Just to mention here one location, genotypic error, genotypic variance. We have here a different location, but the assumption here, what we are trying to say with this personality, is that this is not exactly the same. The genotypic error or the genotypic variance here is not the same that I have here. It's not the same that I have in this other location and this. So different genotypic variances, but also we cannot, let's say, assume that we have the same genotypic error, the same residual error. So this is basically the concept of personality for each location. There is also something important, which is the imbalance. Sometimes we start with a population, we select some promising varieties, or we drop some of them because they are the worst one, etc., etc. So this causes that the genotype by environment classification is incomplete. We can basically see here, for example, in the first year in 2013, we have this amount of genotype. We remove some of them, we add some others, and in that way, at the end, we are gonna have this imbalance, and we have to basically to consider that when it comes to the analysis. And due to selecting, promising genotypes and dropping the rest from the trials in the sequence years, as well as due to adding new genotypes or testing just a portion of the genotypes in certain locations. So this is basically the reason why we have this imbalance in the genotype by environment classification. There are some other difficulties. So the amount of data can be large, with difficulties in fitting and convergency. So, we have a lot of coefficients, a lot of parameters. We have to take care about each experimental design in each location. So it increases the amount of coefficient and et cetera, and makes them all difficult to converge. So we need to take that into account. And sometimes we end up with convergency problems. That's basically the reason. So just to avoid a little bit that, we need for sure require additional things. Exploratory data analysis is something that we must do every time we are going to analyze something like this. Start by checking the genotype names. That's very common that we find this mistake, because in some locations I have a genotype with double space, or I have in a different location with just backslash or something like that, or capital letter or lowercase, etc. So we have to make sure that we have cleaned that to be able just to analyze our data set. Is filter strange or streams observation is something important. We saw in the previous session that having just one or two stream observation can cause damage, in our data, in our estimations, heritability, blobs, blues, etc., etc. So we have to make sure that we have at least process or clean the data before jumping to the MET analysis. So carefully cleaning processes, as I mentioned, start analyzing every site individually, determining all necessary and significant design effects and error structure, etc., etc. So every single site individually and every single trade individually, you know. If we have more than one trade, for sure we need to do that for all of the trades. So this is just like the strategy for fitting MET models. So evaluate which sites to consider for full analysis. You know, we always have to analyze every single site, as we said before. Check, for example, the editability. What is the editability of each trial? And then if we find something strange, I don't know, a poor editability in one trial, we can think about removing that particular trial. We have defined, or we can find in literature that people used to use 0.2 as a threshold. I mean, if we find something lower than 0.2, we can remove it, or let's say omit that tray and then analyze the rest that have a higher editability. And then it's basically combine all the trials into a single analysis and progress slowly to more complex variant structures. We are gonna see that just now. But just something that I mentioned in the previous session, it was about the blobs. And it's because there are two approaches for fitting MET. I mean, there are several ways, but the most known are the one-stage approach and the two-stage approach. The one-stage approach is the one that combines everything in one single model. The two-stage approach is the one that we basically analyze trials one by one and then combine everything in the second model. But how can we go from the one stage to the two stage? So we have to make sure that we are able to understand the blue and blobs. The blues is the one that we are going to use in the second stage as a response variable. So we have to estimate the blues, and also we have to save the standard errors just to estimate the weights. And in that way, we are going to be able to estimate blobs across, for example, locations. Blue, basically the median, is best linear unbiased estimation, and blob is the best linear unbiased prediction. Best is because it has the least variance, and linear because the prediction and estimates are linear function of the observations, and unbiased because it's an estimation of prediction that has no systematic error or bias. That means that the expected value of the estimate is equal to the true value. I mentioned that previously, and it was basically that we have a shrinkage, and that shrinkage depends on the genotypic variance and environmental variance. That level of shrinkage, that is basically the editability. And when it comes to analyze the data in a two-stage approach, we don't wanna like a double shrinkage our estimations. So that's why in the first stage, we have to estimate the blues, and in the second stage, we are basically able to estimate the blobs to avoid basically the double shrinkage. Here's basically what I mentioned in the previous session. The one stage, simultaneously modeling of the within and between trial variation, and it uses all the information, the raw data at the level of plot. But it can quickly become complicated, as I mentioned before, if we have many trials. So we need to take into account all the coefficients, all the variance components that we can have. But the two-stage, I mean, it is divided, as its name says, basically divided in two stages. The first stage analyze every single site individually, just focus on modeling the experimental design, you know, and then in the second stage, I mean, in the first stage, we have to save the blues, we have to say the standard errors of the estimation, and then with those standard errors, we are able basically to estimate the weights, which is basically 1 divided the square standard errors. And we use that basically to connect the 2 stages. It's simpler, pragmatic approach with no or little loss of information. So the focus in the second stage is on the G/E interaction.
And Johan, could you please explain a little bit, like in the first stage, like they are going to be analyzing their experiments and there is going to be a column that is called the weights. Can you explain just a little bit why did we included that?
Yeah, perfect. So, in the first stage, we need kind of like a pass the uncertainty that we have for those estimations. So the way we use is basically, or the way we can use to take that into account is basically use those weights, which basically contains the standard error of the estimations of the first stage. So somehow we try to connect them. I mean, just to, don't lose of information that it says here. If needed, the appropriate weighted, it can be used to carry information from one stage to the second stage. We will see the example in a minute. I have to skip this one because it's the one that we saw previously. But this one stage, in the first session, we learn a little bit how to analyze the single trials. We saw CRD, RCBD, alpha-lattice, row column, etc., and SPATS, the approach to model. And this is basically the first stage approach,
There is a question in here. Which one stage should I remove the outliers? At one stage analysis?
Okay. Yeah, in the first stage, when we are analyzing every single site, we need to check strange observations, and then also to estimate the heritability for that particular trait and that particular trial. And then if we see that it has a poor heritability, we can think about omitting that trial and then analyze the rest, just omitting the one that has the poor heritability. So that means in the first stage. But we can also think about, trials that are totally strange. We can also omit them, because if they are, for example, totally opposite to the rest, so we can think, but what is happening to this particular trial? So we can think about omitting that trial, that particular trial. So it can be also seen as an outlier, a trial which is basically totally strange. And this is the first stage, completely randomized, etc., etc. Once we fit our model, we save the blobs, and we also estimate the heritability, etc., etc. Then we can jump to the second stage. We showed basically the advantages previously, that the spatial approaches can help us to improve our genetic estimation and also to improve our heritability estimation, etc., etc. So this is mostly the reason why we implement the spatial analysis into MrBean, because they have some particular advantages. So what we are going to see on MrBean is that in the first stage, we are going to analyze the trials using the spatial approach. We mentioned this. It doesn't matter the randomization we have. We can assign, or we basically can organize them in the XY axis. So basically we can assign row and column information for the rest of the designs. So if it was originally created as a alpha-lattice design, we can easily recover the information of the row and column. And then we can analyze the alpha lattice design as in spatial or row column design. In the first stage, we take care about that, and then in the second stage, the focus is on the genotype by environment interaction. So here is where we can see or find different variance covariances to model this, depending on the assumption we have. This is the compound symmetry, the classical genotype by environment. The diagonal, every single side is going to have a different genotypic variance, but there is no covariance. It's not a very good or a real scenario. The factor analytic and the structure, we will see then in much more detail in a bit. What is, I have been basically mentioning these variance covariances, but we have to make sure that we are on the same page. Variances, as I mentioned, we have different genotypic variances, we have different genotypic errors, but this is basically telling us about the dispersion, how much variability we have. We can see here, okay, if I got a variance of 100, so that means that we have more variability or more dispersion, etc., etc. So this is variance, but if we think about covariance, we can think about, just for a bit, about the correlation coefficient. That's something that we are very used to. We talk about correlations here, 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 but inside the correlation, we have the covariance. So basically, if we are able to estimate the variance at each trait, for example, if we are able to estimate the covariance, we are able to basically to estimate the correlation, because the covariance is inside the equation. So what if I got a covariance of 0? That means the correlation is going to be 0. If I got a covariance, a negative covariance, that means that my correlation is going to be negative. If I got a positive covariance, that means that I'm going to have a positive correlation. And, that's the reason why it's important to think or to learn a little bit more about the variance covariances, because it helps us to estimate correlations. So this is the identity variance covariance. The assumption here is that all of them, I have the same genotypic variance or the same genotypic error for all the experiments. It's not real, but also that doesn't exist a covariance between the experiments. There is also a different structure, which is called diagonal, as I mentioned. This one is a little bit more flexible. It has different variance or genotypic variance for all the locations. But the problem with this one is that it doesn't have, this covariance between locations. So there is one which basically takes care of this, and it is much more flexible because it has not only a different genotypic variance for all the trials, but also different covariances between each pair of trial. So this is the one that we basically want to go when it comes to, genotype by environmental modeling. But the number of parameters require, parameters, or basically the ones that we don't know, and we try to estimate. With this structure variance covariance, I mean, it's kind of difficult to estimate a lot of parameters, especially when it comes to big or a lot of number of locations of environments. So the number of parameters required to fit fully specific multi-environmental trials increases faster than the number of environments. So that's why we need something more parsimonious, or prefer when the number of environments is large. And there is one variance covariance structure that nowadays has been becoming like more famous, to basically capture the genotype by environmental interaction. Because the point with this one is that this VCUP doesn't require extraordinary model complexity, which makes it like quite useful. This is, the factor analytic structure. This is the UNE structure. This one looks a little bit more complicated, but it's not that complicated. And it has less parameters than this fully UNE structure, variance covariance structure. I'm gonna stop there. I don't know if there
is a question in the here, Johan. What is the minimum trial numbers to model for the factor analytics 1 structure?
What is the number?
Minimum trial numbers.
Okay, but let me check. There are some questions. I'm just checking the chat here. What is the minimum trial number to model with factor? Okay. The minimum trial number, it says factor analytic 1. We can have also more orders, of the factor analytic. We can have factor analytic 1, factor analytic 2, 3, 4, 5, etc. And if we have 2 locations, we are able to estimate, for example, a factor analytic 3. It's always one number less. I mean, we have to have 3 locations to estimate a factor analytic 2. We have to have 2 locations to estimate a factor analytic 1. It's just like the way it works. Santos has a question. Thank you.
Yeah, it's a very nice presentation, Johan. My question is, in your experience, what is the minimum number of genotypes to test in all the locations to capture the genotype by environment effect and the genetic correlation across locations?
This is a very good question. And it can be controversial sometimes because some people, and we can find in literature and also the ones that have written these models, they say that 20 is a decent number to estimate these parameters, to estimate these variance covariances. But I have seen some simulations, but to be honest, the number is even higher. But for sure, I mean, if we are able to have 20, we are going to be able to estimate at least something like decent, So it's like the magic number there. Yeah, let's jump for a minute to the example. I'm gonna close here. And I'm gonna do the example here with this. I'm gonna refresh this. As I mentioned, this is the repo, MrBean app. The way we have to follow to install this is one of these code here. We can use remotes, or we can also use DevTools to install basically the package. And once we have the package installed in our computers, we have to copy these 2 lines in our R script, and then we are going to be able to basically open the interface. Here I have basically the options. People can also create an icon in their desktops if they don't want to go to write those codes in the R script, etc., but you are going to be able also to do this. And if you need my help, please don't hesitate. I will share with you the script and just to how to generate it. But okay, here is the interface. I'm going to show you the process of fitting the 2-stage model. Yeah, I have to make emphasis on this. It's basically that we have available here the 2-stage approach. We don't have the first stage approach due to the complexity. But here we have to go to this import data, and I have a data set here where I have 12 locations. But just I'm gonna analyze some of them. I'm gonna analyze. We can make a subset of the data set here just by clicking subset variable, and we have to select basically the levels. I'm gonna select these 4 locations just to illustrate this. We can jump to descriptive analysis if we wanna see, I don't know, plot the box plots. This is the response Data set is the variable that defines each individual trial. We can see here the location DAR16, DAR16C low phosphorus, medium phosphorus, and this is a location in Palmyra. Here we can see the distribution. We select the response variable, and then we are able to see what is the distribution basically of this particular trait. And we can also visualize some summary statistics, mean, standard deviation, minimum, maximum, etc., etc. But also the plot helps us to see that, right? Also these descriptive statistics that appear here. And once we are happy with the data, we can jump to the side-by-side analysis. We can also go one by one. I mean, just subset the data set for one trial and analyze it completely in very much detail, but we can also jump to the side-by-side. We have to select the yield, the response variable. In this example is the yield. The genotype, we have the genotype as a line here. Data set is the variable that is defining the experiment. And we have to select the col and row. All of them have been randomized as an alpha lattice, but we have been able to recover the row and column information. We can see here if there are some missing data for this trait and the amount of genotypes. Here I can leave this empty if I want. If I want also add some additional fixed factor, random factors, etc. And this is the R limit, a numerical value used for determining when a value is considered an outlier. So we have set up this as a 3 by default. But if you want to be more strict, we can also play with this slider. Continue.
And, Johan,
Yeah.
before you continue, just one clarification in here. So you have the click for the random genotype. So can you explain in which situations will you consider the genotype as random or the genotype as a fixed effect?
Perfect. Yeah, that's a good point. First, if I want to explore the genotypic variation, heritability, and also remove string observation, I can leave this as a random. And that's what we are going to do now. I want to see the heritability and genotypic variance, etc. And then, I'm sure I got, for example, good heritabilities, and then I will be able to feed the MET. But yeah, this is just for checking, estimating BLAPs, if the case. But if I'm going to estimate a two-stage approach, I have to change this checkbox. And this is basically telling me that I'm gonna get the blues. And also it will help me to estimate this weight to be able to analyze the data set with the two-stage approach. But I'm gonna leave that as a random just to check what is the heritability, And then I can see here what is the connectivity or the amount of genotype share between locations. On the diagonal, I can see the number of genotypes tested, and out of the diagonal is the number that basically share. And I can also explore in much detail the connectivity. I can see, okay, this genotype was present just in this location. This genotype was present just in this one, and like that. You can see here that this one was present in 2, but this one in 3, and this one in 4 locations. So this is what we were mentioning, basically, that we have this not completely balanced genotype by environmental classification. We are happy with this, and then we can basically run the model. I don't want to run the 12 experiment because it can take a while. So I'm just gonna use this 4 experiment to see what is the output. What it's doing right now is basically fitting an SPATS model for each trial for this particular trait, which is the yield. And we have to wait a little bit while this is running.
Yeah, I can be telling you like this is a testament as a user. Like in the case of MrBean, it's really, really fast, which, like this type of task used to take hours, 24 hours, sometimes more. It takes only seconds. So that's really cool.
So yeah, thank you. And now that we have basically run the model, we are able to see, okay, what is the genotypic variance? What is the residual variance? Compare them. And here on the right side, we basically can check the same plot that we see or we saw in the single site analysis. So if we click on this experiment, we can go, through the different experiments. You can see here the fitted spatial trend. We can also explore this. This experiment has a lot of spatial variation. We can also see here, this one has a pattern in the field, and etc., etc. This is a good example because I don't need to have, the layout doesn't need necessarily to be like a square or something like strict like a square. It can have portions, like a square or rectangle here, etc., etc. And the model is still able to converge. We can explore the summary as well. The summary of the fitted model. Here we can see the ranking plot. We can see here, we can make some to the plot here. Okay, this is the best one, etc., etc. We can see what is the worst one. This is just the genotypic blob. Is the blob, so that's why it's centered in 0. So yeah, don't scream because we got negative values. It's just because we are visualizing the row blob. On the left side, we can explore, this is a Pearson correlation. On the diagonal, we can explore the heritability for the trait. This one is very poor, for example, 0.21. The other ones are quite good. And I can see that there is, they have a positive correlation and it's significant. I can also explore genotypic variation, genotypic error variation, heritability. And as you can see here, there is no potential extreme observation. The R squares and the CB, the coefficient of variation as well, are able to explore, the quality of the data. Here we see the predicted values and standard errors by each genotype and by each experiment as well. And, I cannot see here the weight because I am estimating basically the blobs. And the idea is that once you fit the model and when you click on this checkbox and define the genotype as fixed, then you will be able to see the weights on this box. Here we can also explore the outliers, but in this case, we don't have streams observation, so we are not going to see something here. So let's assume that I already run all the information. I already run the 12 experiments. So what I'm going to do is basically download or to take the file that MrBean provides me, and I'm going to import this one. If you want me to show you this, what is the format it has to have? It has experiment, genotype, predicted value, standard error, and weights. You can also, if you have your, I don't know, RCBD or different experimental designs, but also you have saved the standard error, you are going to be able to stack all the experiments and you are going to be able to use this module as well. So we import the information here. We define the response variable, in this case, the blues. And then our genotypic column, line, experiment. If you wanna make a subset of them, the column that has the weights and the variance covariance structure. There are several variance covariances that we can fit. We can explore here, okay, core H. What is core H, for example? What is core GH? Is basically the one that I mentioned in the presentation, the unstructured one, the factor analytic one, factor analytic 1, 2, 3, 4. So we can start with one very simple, for example, the diagonal. You know, here is basically showing me the number of genotypes by site, how they are connected. I can explore here the connectivity between them. And also I can explore the means. Okay, this one has a higher means, lower means, etc., etc. And then I'm gonna be able to run the model. I can see here, this is the model, the number of variance covariance components, the log likelihood, AIC and BIC, just to compare different models. As you can see here, it's quite useful also to play and to understand the different variance covariances that they are available here. Because, okay, I can see here, okay, the diagonal variance covariance is basically assuming that every single side has a different genotypic variance, but has no covariance. Okay, that's why I cannot basically see something here. It's basically 0. But I can also play with some others, you know. Assume that I have the same genotypic variance for all, but also the same correlation. I'm gonna use this one and I'm gonna run this model. I can explore this one. And you can see here on the diagonal is exactly the same value, and out of the elements out of the diagonal is basically the same value repeated. And the correlation is the same for all the trials. This is the classical, the compound symmetry model. But you know, the assumption is not that real. So we can explore something more complicated. For example, different genotypic variants and a common correlation between trials. Let's define that one. And then I can also check this one. Here, I can see that on the diagonal, I have different genotypic variances. And out of the diagonal, I can also see different components, but the correlation is the same. But what if we start estimating, for example, the factor analytic ones? So this is more complex. And the point with this one is that the correlation is going to be also different. So some of them are lower, some of them are higher. And here, everything is basically different. Every single site has its unique genotypic variance. And every pair of trials is going to have different covariance. So with this one, I can explore here the covariance, the correlation. You know, I can also explore by creating this nice plot to see if there is something like behind the correlation. I can see here that there is something like a strong dividing these trials. And it's basically the location. This PAL is because it's a location here in Colombia. It's Palmira. There is a different location, Darien. And here we can see Quilichao. But Quilichao is more similar to Palmira, what the data sets, you know. And Darien is a little bit different. We can explore this. And if you want to see what is the equation or basically the the structure of the model, we can also explore it here in much more detail. What is basically what we are fitting or what MrBean is doing behind. This is the plot that ASRML generates. And Here we got the predicted values. We can see the overall predicted values across locations. You know, this correlation 0.46 is not that bad. It's not something negative, so we are able to estimate something across location. But for sure, if we want to go to separate the trials, I mean, just that end, to estimate that nominal breeding value, we can do that also. But what I see is that the correlation is very positive or highly correlated. So I would basically end up with an overall breeding value for selecting. There is a model here to explore using this principal components analysis. I'm gonna remove the points. Rabbit. We can see what are the best ones. All of them are positive, so the arrows are going to the same direction. The best one to this side, the worst one to the other side. Here we can see the variables, in this case the trials. We can also explore the most extreme ones. Okay, this one, etc., just to explore this genotype by environment interaction. There is something here which basically can help us to, let me check, to explore in much more detail. As we said before, we have different genotypic variances, and we have also a variance explained by the components. We are fitting a factor analytic tool, so that's why we see here two loadings, two components, factor analytic 1, factor analytic 2, and there is a percentage of variation explained by them. The blue one is related with the first factor, and the other one is related with the second factor. And if we want to see what is the average in total, like the percentage of variance explained is quite high, you know, almost 90%. And the plots that we saw in the PCI are very similar with this. Here we have some scores to explore the genotypes, and here we have the loadings to explore the trials. So the ones that are closer is because they are more similar than the other ones that are like separated by longer distances, greater distances. Here is something that if you want to explore more detail is the latent regression that we can also explore using the factor analytic approaches, and we can compare genotype by genotype. We can define here, I don't know, the best one with the, I don't know, sir. Let me check this one. If I want to compare them, basically I can compare both. If I want to use the labels just to see, okay, this genotype is best performance in this location, this is in this direction. And
so
it
this is very similar to the stability analysis, correct?
Exactly, yeah, it helps me to explore also the stability of the genotypes. And once, you know, I'm happy with this and I want to just basically download the overall breeding values, I can go to this one and download my overall breeding values, download the genotypic correlations, download the variance covariance information. And also these plots, we can also download it. So on. There is also a point that I don't know if we have enough time, but I will go very quickly to this genomic prediction side.
We have 9 minutes.
Exactly. I will take 4 minutes just to explain a little bit this one. You know, this is also very famous methodology, the FielDHub, where we basically combine the phenotypic with the genotypic information. So I have an example. Just let's assume that I already analyzed my data. Let's say that I analyzed all these trials and I got the overall blues, but I also have the genomic relationship matrix, and I'm gonna basically combine it to estimate the FielDHubs. And in case I have more genotypes than phenotypes, I can also predict the ones that doesn't have phenotypic information. Here, I'm gonna import the phenotypic data. I'm gonna add the genotypic information here. Here in this genotypic data, it has a numeric format -101. And also, if you have the genotypic map, you can also import it there. For example, you add the markers and also the map. You can also import it here, and we will see the information right here. The data imported is genotypic, color, yield, etc., etc. And here we can see the genotypic information on this one. I have this amount of markers, this amount of individuals, and let's say I wanna estimate the G-block for yield and also color, and I'm gonna select the genotype. The current method available is the G-block. Summary is the package that is using behind, and we can see here, okay, yield is more complex, so color is simple trait, so we can see here the differences, but also we can explore here the heritability, the genomic heritability using marker data. And here is the summary. The genomic heritability for color is higher than the yield. And we can explore basically the accuracy. Yield has less accuracy than this color trait, which is the simple one. And I can download this table if I want, but I can also explore, this, let me show you here, the correlation between the traits and also the genomic heritability on the diagonal. If I want to explore in much more detail the predicted values or the G-blobs, we can see here, okay, what is the correlation between them, especially if I have more than 2 traits for sure. And, I am able to estimate the GEVBs, predicted values, the ones that doesn't have phenotypic information. You can see here, this genotype doesn't have or didn't have phenotypic information, but I'm able to estimate it, because of the genomic relationship matrix. And I can also check the reliability of the estimation. If this is close to 0, it's because it's less reliable. But if I have something higher, it's because I have more, it's more reliable. And at the end, I also I can see the marker effects. So each position in the genome is going to have an effect. So this is what people have called GWAS by G-blob. So we are going to be able to explore this in much more detail. So here we have the yield and color. Basically, with some math, we do some math behind with these G-blobs and also the marker effects, and we end up with this. These are the estimated marker effects, and we are able basically to explore like we usually do in GWAS analysis, exploring the peaks and also, try to find something in the genome. We can also download the markers if we want to, for example, use them to estimate a new genotype. I don't have phenotypic data, but I have the markers. I'm going to use the markers to estimate that one. That will be the way we can basically follow. This is the prediction plot. Here is the one that have been fitted. This is the one that have been predicted. If I don't want to include the predictions, etc., etc. So I think we are running out of time, Anna, I think it's time to.
Yes, I think that we can maybe have some few questions. One of the questions was if MrBean has available to do a trade by trade within the MET analysis.
Sorry, what did you say again?
The question is like, does MrBean has the trade by trade analysis in the multi-environmental trial analysis section?
Let me check.
So can you analyze more than one trade at a time in the MET?
Yeah. In this case, It's just one trait at a time, you know. You first analyze one, but that's more or less the focus. Is not that flexible, but in the future it will be integrated. I mean, to be able to analyze multiple traits in the multi-environmental module. Santos said, in my experiences, but before running the spot mode, I need to make sure that I had row and column not repeated across. Oh, that's a good point. Santos mentioned that. Luis, I didn't know I could drag and drop the CSV. Yeah, you can do that. Yeah, because if you have your, your folder open, you can just click on then and drag on the bar. So it can be also do it. So I don't know if there are more questions. So I think we can finish here.
I don't see any more questions on the chat. And I just quick mentioning, like we have been posting in the chat a little bit of some different resources, like how to download FielDHub, how to download MrBean, and as well as kind of like a companion papers that we have been utilizing to put together this presentation. So thank you.
Okay. Hello, everybody.
Hello.
I think if we can either hold off for a few moments to take questions on the chat, but if not, I think we can just thank the group at NDSU, Anna, Johan, and Didier for the wonderful presentation. Okay, we can give you a round of applause here virtually. There we go.
Thank you.
Thank you, everyone. Yeah, thank you for joining us. It was our pleasure.
Yes, thank you, NAPB, for making this possible. We are really excited. So
Always glad to have good speakers like yourselves. All right. Well, I think with that we can